Hi, everyone. Welcome to the Assay Guidance Workshop for High Throughput Screening and Lead Discovery. We are really excited to have you all join us today. I am Sareen Markosian. I'm the new editor-in-chief of the Assay Guidance Manual, and I will be moderating the workshop today and tomorrow. So a couple of announcements before we start. The first one is that we are on Twitter. We have a Twitter handle, and you can follow us at Assay Guidance. Today's workshops has a hashtag, and that hashtag is #AssayGuidance2020. So please feel free to tweet about this event and use that hashtag freely. Next is I would like to ask the panelists to make sure they remove their virtual backgrounds because WebEx has issues and the camera freezes if you use that. And then also disconnect from your VPN to ensure your talk goes smoothly. Okay, with no further ado, I would like to introduce our next speaker, who's Dr. Christopher Austin. Dr. Austin is the director of the National Center for Advancing Translational Sciences at NIH, and he will be giving the welcoming note. And please, just an announcement for this, increase your volume so that you can hear Dr. Austin properly. Thank you. The floor is yours, Dr. Austin.
Good morning. My name is Christopher Austin, and I'm the director of the National Center for Advancing Translational Sciences at the National Institutes of Health. And it's my pleasure to welcome you to this Assay Guidance Workshop for High Throughput Screening and Lead Discovery. I'm going to tell you a little bit about NCATS and translational science and where the AGM came from. But before I do that, I want to give you some up-and-coming events that are happening on the AGM calendar. In addition to the workshop going on today and tomorrow, we'll be having during 2021 focused workshops on some new topics, including antibody therapeutics and DNA encoded libraries. You'll be hearing a bit about those later in the program as a teaser. And also we'll be having a workshop early in 2021 on repurposing for COVID-19, and you'll hear a little bit about that from Matt Hall at the end of tomorrow. Lastly, SLAS Discovery is planning to publish in 2021 a special issue entitled The Assay Guidance Manual for Drug Discovery, Robust or Go Bust, which has become a bit of a tagline around here. And it's going to be focused on the Assay Guidance Manual and the workshops that have come from it. We've successfully solicited 14 manuscripts for that issue so far, and I hope you'll be on the lookout for that. So let's dive in. Let me just bring up my slides here. So again, welcome. I think you're going to learn a lot over the next 2 days. Every time we do this workshop, we get great feedback from the participants, and I hope you will give us feedback on what worked and what didn't, and what you want to hear more of or less of at the end of the workshop, because that's really important for us for planning future workshops. So the word translation is widely used in the biomedical vernacular these days, but it's used in different ways by different people. So I always want to define how NCATS uses this term and some related terms. So in NCATS vernacular, translation is the process of turning observations in the laboratory, the clinic, and community into interventions that improve the health of individuals and the public, from diagnostics and therapeutics to medical procedures and behavioral changes. And notice what's in red, turning observations into interventions. And when observations are made for the first time by a researcher in the lab, or a doc in the clinic, or a public health worker in the community, those may not be robust and reproducible findings. And so the first step in turning those observations into interventions is to find out, well, just how robust is this observation? And you're going to be hearing a lot about that today. all of us probably use this related term, translational research. We refer to translational research as the endeavor to traverse a particular step of the translational process for a particular target or disease. And many of you on at this workshop will be doing this in your career, and it's a very widespread and important discipline. But translational science we might think of as the first derivative of translational research. That is, translational science is the field of investigation which seeks to understand what the general principles are, scientific and operational principles that underlie successful translation at each step. I think all of us who have been involved in this endeavor during our careers realize that this is an almost relentlessly empirical process. And if one has an increasing number of degrees of freedom as one goes from a protein in a tube to a person in the community, and a multi-step process with a low yield at each step, the likelihood of your getting from successfully from the beginning to the end approaches zero. And in fact, that's exactly what we see in therapeutic development. And so NCATS is focused on on turning this process from an empirical phenomenological process, indeed, to a predictive science. And that's part of what you're here today to gather. So the NCATS mission, in a lot of fancy words, it's to catalyze the generation of innovative methods and technologies that will enhance the development, testing, and implementation of diagnostics and therapeutics across human diseases and conditions. But I want you to look at the words in red here again: innovative methods and technologies. And again, this is what NCATS is all about: new ways of doing things, and that's what this workshop is about. So there's a lot of translational problems that prevent efficient translation of a fundamental discovery to an intervention that improves human health. But a number of years ago, we put together, like any good biochemist would, a list of the rate-limiting steps to this, if you will, biosynthetic pathway. And these are the problems among them. These are the major problems that bedevil the translational process, no matter what disease or target you happen to be working on. And this workshop will focus on certainly the understanding of translation, but in large part the left column here on this list. But I also want to point out that a lot of the work that NCATS does is more perhaps in the organizational or cultural domain, particularly in the academic world. And you'll notice their education and training very prominently second from the end there. And again, that's why you're all here. So rigor and reproducibility. You will all remember that almost a decade ago, there was a fairly major cataclysm, at least in the academic world, to publicize something that all of us who had worked in pharmaceutical companies or biotech companies already knew and had built into our processes, which is that the vast majority of academic discoveries that come to drug development organizations are no-goes because they're simply not reproducible, and so they're quickly triaged and go nowhere. Now, this is an enormous waste of time and people's resources and money and people's careers, and so appropriately, NIH has been focused on a number of things to improve rigor and reproducibility. But this is a particular problem in translation. It's a problem across biomedical research, of course, but it's a particular problem in translation because, by definition, it requires the practice of a discovery made in one lab by scientists in another. And so what that means is that the famous postdoc magic hands, or the graduate student who happens to be able to make an experiment work, just doesn't cut it in translation. the standards are much higher for rigor and reproducibility, and they have to be. Therapeutic discovery programs have to be built on robust findings that cannot be operator dependent and can't be affected on by small changes in experimental conditions. And so what you'll learn about over the next 2 days is part of the answer to this problem. So what translational rigor, how do we define translational rigor? It's a saying which we instantiated here at NCATS almost 2 decades ago now and has served us well, which I would recommend to all of you, is the attitude that every finding is an artifact until proven otherwise. This is empirically observed to be true, and there are artifactual explanations for virtually every finding that vastly outnumber biologically meaningful explanations. And it puts the burden on the investigator, on the project scientist, to demonstrate that finding is robust and reproducible, not on others to prove that it isn't. And this is very important and results in more efficient translation because there's fewer dead ends. So this is really where the Assay Guidance Manual came in. This is a wonderful story that perhaps you'll hear about over the next couple of days. It started with a public-private partnership between a predecessor to NCATS, the NIH Chemical Genomics Center, and colleagues at Eli Lilly, taking their internal assay development manual and screening manual and putting it on our website, the NCGC website at the time. And this very quickly we learned that this fit a very important need in both the academic and the biopharmaceutical world for knowledge and detailed protocols to work in the early therapeutic development space. And so over the years, we've turned this into an electronic book with dynamic updates, which of course it is now. And it provides not only background and rationale, but detailed protocols, step-by-step protocols for translational methodologies that in fact are robust and reproducible and consistent, and whenever possible, high throughput and automatable, and importantly, biologically and translationally relevant. And for those of you who are of a certain age, will remember, like myself, will remember by analogy the Maniatis molecular cloning manual. This is how I learned how to do molecular cloning, and then more recently, of course, current protocols in molecular biology, which spawned a large series of current protocols, which have served these purposes in the academic world. But before the Assay Guidance Manual, there was no equivalent for early drug discovery, and I think that's why it's filled such an important need. So lastly, what is an assay? We're going to be talking about assay guidance for the next 2 days. So what the heck is an assay? Now, assay, of course, just means try in French, but in biomedical terminology, it's an analytical method, a measurement procedure defined by a set of validated reagents. You notice each of these are underlined because they're stress their importance and of the robustness and reproducibility of each of what's underlined here. Validated reagents and reaction conditions that produces a detectable signal, allowing a biological process to be quantified using appropriate data analysis procedures and models. And you're going to be hearing about various approaches to these assays. Of course, almost everything we do in the lab is an assay of one sort or another, but not all, in fact, most do not meet these really stringent standards that you're going to be hearing about for the next few days. So again, welcome. We're glad you're here. We hope that you enjoy the next 2 days. I know you will, because I've listened to all of these presenters before, and I think you'll really both enjoy a lot and learn a lot from these. And we've tried to build into this some time for discussion as well and questions, and I hope you'll take advantage of that as well. So welcome and have a good next 2 days.